Hello U-turns, we're about to do your biology crash course. Everything that we've covered since the beginning of the unit is going to be in this topic. The PowerPoint will be up for you to download and take notes in later. Make sure you have something to drink, something to eat with you. There will be breaks in between the video, between each topic, so take a break, check your notes, check your understanding, check the PowerPoint, and because it's recorded, you can watch it as many times as you like. You can comment underneath if I've missed something. And yeah, alright, let's get started. Right, year 10 biology, let's go. Alright, so starting off with DNA. Couple of fun facts. Chromosomes are made out of DNA. DNA stands for deoxyribonucleic acid. So molecules stores genetic information in cells and copies itself exactly for new cells. Alright, so DNA, deoxyribonucleic. That acid is often called the blueprint of life. In simple terms, DNA contains the instructions for making proteins within a cell. Alright, so proteins are responsible for all cell structures and functions. They're made of long chains of amino acids. There are only 20 different types of amino acids in the human body. And proteins are responsible for literally everything: hair, skin, hormones, muscle movement, antibodies, chemical reactions, oxygenation of cells. All of it, that all comes from proteins. Right. Next, so DNA, structure of DNA. DNA is a polymer. So a polymer is something that's made of repeating subunits. Also, the repeating subunits are called monomers. In the case of DNA, they are nucleotides. So they're two long strands right next to each other, and each is a chain of nucleotides, like this here. One chain, two chain. Alright, so nucleotides consist of a phosphate, a carbon sugar, in this case the sugar is called deoxyribose, and a nitrogen base. So you have your phosphate there in blue, your pentose sugar, and then your nitrogen base. So when you are asked to draw a base, you need to draw the phosphate, the sugar, and the base next to it. Phosphate has to be round, sugar has to be a pentagon, and your base can just be a rectangle. Okay? Okay. Same thing, different way of showing it. Phosphate, sugar, base. So each nucleotide is made of 3 parts. Your 5-carbon sugar, that's why it's a pentagon, because there's 5 carbons to make that up. Your phosphate group, which is here on the side here, and your nitrogenous base there. Okay. So that there is a single nucleotide. That's your A. So the backbone of DNA is formed by alternating sugar and phosphates held together by a strong bond. So sugar, phosphate, and it just keeps going all the way down on both sides. And in between here, we have hydrogen bonds. So there's 2 between A and T and 3 between C and G. Okay? The rungs of the ladder are formed by the 4 nitrogen bases, and they're held together by weak hydrogen bonds. There. Okay? So, A is for adenine, T for thymine or thymine, C for cytosine, and G for guanine. Okay? A, C, T, G. If you want to have a look what they look like chemically when you draw them as a chemical complex, A, C, T, G. Alright. Same information, different way of visualizing it. Some people draw their bases like this. So adenine has a curve, guanine has a triangle, thymine, triangle, thymine, curve, cytosine, triangle. But these parts here, the chemical structures, are the same. Okay, so the order of these 4 bases here determine your genetic code. Alright, so DNA strand, each nucleotide bonds to the one to form a next strand. 2 strands twist around a central axis to form a double helix. The sides of the ladder alternate between a phosphate and a sugar, and the rungs are held together by hydrogen bonds. So, phosphate, sugar, adenine, thymine, cytosine, and guanine. Hydrogen bonds in between the middle here. You can see them faintly. Okay. Next. So, DNA bonds by something called the base pair rule. So, A only bonds to T, and C only bonds to G. That's the base pair rule. You won't get any other combination for DNA. RNA is a little different, and we'll get into that in a bit. Alright, so nitrogenous bases. These 4 bases have endless combinations, just like the letters of the alphabet can combine to make an infinite number of words. The 2 strands are said to be complementary because, they match up and they complement each other because they're the same shape. So that means if you have GAATAC on one side, you will have, and you have a blank spot to fill in here. So pause it, fill it in. And the answer you should have is CTTATG. Right. Replication. So DNA replication is the process by which DNA makes a copy of itself. Why do we do this? So cells divide for an organism to grow or reproduce. Every new cell needs a copy of that DNA. So this DNA replication is part of mitosis and meiosis, which we are covering later. So in DNA replication, enzymes work to unwind and separate the double helix and add complementary nucleotides to the exposed strands. So we can see here, this is the old DNA strand, and it's become unwound here. So the old strand is continuing down, and the new strand is building upon that. Okay. So this means DNA replication is semi-conservative. So it means you have some of the old strand, and the new strand is added into it. So when it makes a copy, one half of the old strand is always kept in the new strand. This helps to reduce the number of copy errors. Okay. So with DNA replication, so we have DNA helicase. It breaks the hydrogen bonds that are here between the 4 bases, and DNA polymerase move along this. They slide up and down the strand, and they add 3 nucleotides to the old strand. So we have the old strand in blue and the new strand in yellow. What's not shown are these 2 enzymes. So DNA helicase will be here and DNA polymerase will be here, just pulling these free floating bases and adding it to the old strand. Alright, so from DNA replication, the result is 2 exact copies of the original DNA. So each new double helix is comprised of 1 original DNA strand and 1 new strand, makes it semi-conservative. Old mixed with new. Okay. So we are moving on to protein synthesis. Time to have a break and come back when you're ready. Alright, so protein synthesis is split into 2 parts. First, we have translation. So we have DNA in the nucleus. To make proteins, DNA must get its instructions to the ribosomes who make proteins. So proteins are made with the ribosomes. To transport its instructions, it uses messenger RNA, mRNA. And even if you're talking about messenger RNA, if it's at the beginning of the sentence, the M here is lowercase to signify messenger RNA. This whole process here, this whole diagram is the whole process of transcription and translation. We will get to that. Okay, so RNA is ribonucleic acid, consists of 1 strand of nucleotides and has a ribose, not a deoxyribose. It has uracil as a nitrogenous base and not thymine. So this here is DNA with its 4 bases, and then this here is RNA with its 4 bases, and there's uracil compared to thymine. Okay, questions. Pause, answer the questions. More questions. Pause, answer the questions. Alright, proteins. So proteins are the workhorse molecule found in organisms. The blueprint for proteins is coded in the DNA of the organism. So whenever we want to make one of these things, we get that information to how to make it from your DNA. Alright, what do we use proteins for? Literally everything. Carrying gases, they're enzymes, they store protein as in your food protein. Movement, they're antibodies, structure stuff. They have so many uses you would not be able to function without them. Okay, so protein synthesis. We have DNA, transcription to make mRNA, to get translated, mRNA, and then we have tRNA here bringing codons to create amino acids on the side here. Right? Very simplified diagram of the entire process. So unlike DNA, RNA is ribose, single-stranded, and uracil, as stated earlier. Okay, so we have three main types of RNA. Messenger RNA, small m RNA, so that carries copies of instructions from the assembly of the amino acids into the proteins from DNA to the rest of the cell. It's called mRNA because it is a messenger. Alright, then we have ribosomal RNA, or rRNA, makes up a major part of the ribosomes, which is where proteins are made. That is the only place you will find rRNA is in a ribosome. And last but not least is transfer RNA, or tRNA. So it transfers amino acids to ribosomes during protein synthesis. Alright, proteins. Revisiting them. Biomolecules. Proteins are made up of a chain of amino acid monomers, and they are formed at the ribosome. So proteins can be enzymes which catalyze and regulate chemical reactions. Proteins also make up our structure. So this whole long thing here, these are all little amino acids. Each dot is an amino acid. And altogether, that long chain is a protein. Also known as the primary protein structure because no folding has been applied to it. But you learn about protein folding when you get into year 11 and 12 if you continue on to do biology. Alright, so 2 steps to make a protein. Transcription, going from DNA to RNA, and then translation, RNA to protein. Like what's happening in this diagram here. DNA to RNA, or mRNA, and then mRNA getting the tRNA to translate its code into amino acids. Okay? Diagram of transcription. So DNA, this section of code is this, and mRNA is duplicating the code, or making a complementary strand of it, for being more correct. So, because we are dealing with RNA, it's a U, not a T here. So A, U, and then T to A. Okay, so it's copying this information here, making a complementary strand, so it can be passed on. Alright, first part of protein synthesis, transcription. So transcription takes DNA and makes it into RNA. So it takes place in the nucleus with help of 2 enzymes. Helicase, which separates the DNA helix, that part there, and RNA polymerase, which brings RNA nucleotides over to be synthesized into mRNA. So the template strand, so that's the strand being copied, so this one here, this outer one here on the left-hand side, is a side of DNA that will be used to create an mRNA strand. The other one, just kind of, we ignore it entirely. So this one here on the left is going to be turned into the mRNA template. Okay, so transcribing the genetic code. Genetic code is read 3 letters at a time. They are called codons. So 3 nitrogenous bases together make 1 codon on the mRNA strand. So the genetic code language from DNA is transcribed into mRNA during transcription, and each codon will have a special name, a special amino acid that matches it. So for serine, we have UCG. For histidine, it's CAC. And for glycine, it's GGU. There is a table somewhere in here that shows that. So this is a diagram of transcription. You need to be able to replicate this diagram here. So you have the DNA unwinding. We have the template strand here, the coding strand. And then we have the messenger RNA. Note, the bases are complementary. And because it's RNA, it's a U and not a T. Okay? Animations, if you want to watch it. Alright, so task in your own words, fill in these questions. You have 2 leading sentences here. If you want them, they are there. Okay, so once we have completed transcription, the mRNA leaves the nucleus and travels to the ribosomes in the cytoplasm. So this is the nucleus, the ribosomes are right here. The ribosomes are the only place to build proteins. You cannot make a protein anywhere else in a cell. They have to be done in the ribosome. Okay, so diagram of translation. This is happening in the ribosome, this loop here. Your mRNA strand here and your tRNA bringing with the codons coming to attach and create an amino acid. And that creates a whole chain there. So incoming tRNA carrying an amino acid here comes in attaches to the chain of mRNA, and then the amino acid joins the rest of them. Okay, translation. Translation is from mRNA to a protein. Takes place at the ribosomes. mRNA that was transcribed leaves the nucleus and attaches to the ribosome. One codon at a time is matched to a tRNA anticodon or anticodon. So if we go back, this part here is the anticodon. This is the codon. When tRNA sits down at its matching codon, the amino acid it carries is dropped off and bonded to the protein chain by peptide bonds. And now we have a protein. So this line, this string here of amino acids is a protein forming. So we have our tRNA here, that square. That green pro in the circle is an amino acid. It's going to bond to this codon here, because it has, so this anticodon is triple G. The codon here is triple C. It will bond, and then this amino acid, the pro, will join that LU, yes, LEU, will join that and be part of the protein chain that's going on there. Okay, so this is how you would draw translation. You do not have to remember what these two amino acids are. You can just write amino acid 1, amino acid 2. If you want to remember the names, you're more than welcome to. Okay, so these amino acids have joined together. This tRNA with its anticodon has joined to this codon here, and it drops off its amino acid to bond and start the chain of proteins. So the tRNA molecule for methionine, that's this one here, has the anticodon UAC, which matches the codon AUG. Okay. You should be able to draw this diagram and explain what's going on. Alright, so this here is an amino acid chart. Used to find out which amino acid joins the protein chain next, and you always read for the mRNA codon. Do not read what is being written by the tRNA. Always the mRNA. Okay, so first position. So if your first letter of your codon is A, you're looking along here. If the second letter is C, you're looking in this box here. And then if the third letter is U, so ACU, you're coding for this amino acid. First letter might be U, second letter may be A, third letter U or C, you'll code for this amino acid here. If your first letter is G, second letter is A, and third letter is G, GAG, you have this amino acid here. Okay, so this is how you read the chart. First, second, third position. Okay, it helps if you have a ruler. Alright, animations, if you want to watch how translation works, also for you to watch. Alright, so, in your own words, purpose of translation, process of translation. Go for it. Alright, an illustration describing the process of protein synthesis. Okay, DNA unwinding, gene being copied, this is mRNA. mRNA leaves the cell, comes into the ribosome, tRNA brings the anticodon to match up with the codon chain down here, and it drops off the amino acid that it's making, and it grows the protein chain until a stop codon is reached, and then we're done. The protein is finished being made. Alright, so DNA replication produces more DNA. When transcription occurs, we get mRNA. From mRNA, we get protein because it's translated through tRNA to make a protein. Right. Process yet again and again. Here we go. You can label. This is for labeling. This is also for labeling. Okay? Questions? Fill in this part. Fill in this part. Break time before you come back. Have a break and then we'll start mitosis. Alright. So mitosis, what is it? Why is it important? And what do you specifically need to know? First of all, the 4 phases that are part of mitosis. Prophase, metaphase, anaphase, and telophase. Alright, so mitosis is cell division. So why do we cell divide? The cell theory states that all cells arise from other cells. How? Because they divide. So why do we need it? So you start it as one cell. Are you still one cell? Obviously not. When you fall on the pavement and scratch your knees, that needs to be replaced. And your stomach acid burns through your stomach lining. Well, it could. How do you contain all that? Through mitosis. So with cell growth, if we split into multiple parts, we won't end up bursting or looking like, what's her name? The chick from Willy Wonka who ate the bubblegum. Violet? Anyway, so we divide, so this doesn't happen. No exploding, no growing to be one massive single-celled organism. Cell repair. Every time a cell dies, they need to be fixed and they need to be repaired and filled in. When you break your bone, you have mitosis happening here, so your bones can be fixed and put back into place. Same with scar tissues. Anytime you injure yourself, you go through mitosis to get your broken parts fixed. Alright, so you always need new cells. So mitosis is a process by which new body cells are formed for growth or repair. This type of cell division results in two identical cells produced from one parent cell. The two identical cells are called daughter cells, right? Mitosis is also reproduction involving one parent and also known as asexual reproduction. Okay? Right. So asexual reproduction happens in single-celled organisms and us. The mitosis part happens in us. Not so much the asexual reproduction. Alright. So mitosis is asexual cell division. It happens in a series of stages. The stages depend on the type of organism, if it's a prokaryotic cell, so single cellular organism, or eukaryotic, multicellular organism. Alright, so stages of mitosis begin with one cell, called the parent cell. The DNA is copied and the cell splits in half. Final product of this type of cell division is two identical daughter cells. The daughter cells are clones of the parent cells. That is important to note. Daughter cells are clones of parent cells. Alright, so mitosis makes clones. Mitosis involves a series of changes that make an identical copy of the original cell. The DNA is copied exactly, which means if there's something wrong in your DNA, it will get copied over and over and over again. So mitosis makes a clone of the parent cell. One parent cell divides and makes two identical daughter cells. Now, going back to DNA. So one of the two nucleic acids you need to know. Why does your cell need it? Because DNA contains your hereditary information. Without this strand, you would not be you. The instructions to make the proteins that make you. So everything so far has been building up to this. So that's why DNA is so important. It contains your genetic information. Without it, you wouldn't be making the right proteins that code for you personally. All your genetic information is stored in these strands. Alright, so DNA is replicated. It must be replicated so that each new cell has an exact copy of the instructions, all your hereditary information. And that's because we are making clones like SpongeBob over here. There's a bunch of him lined up. Okay, so DNA separates and the parent cell divides in two. This division is much simpler, quicker than cell reproduction in eukaryotes. Okay, so we are animal cells. Plant cells are plants. We are focusing on animal cell mitosis. Okay, so we have different types of mitosis. There's budding, vegetation, propagation, and the mitosis cycle in the nucleus us. We are not covering budding and vegetation propagation. No, that's for year 11 and 12. Alright, so with mitosis, one cell becomes two. In eukaryotes, the stages of mitosis begin within the nucleus of the parent cell. The final product of this type of cell division is two new daughter cells. The daughter cells are identical to the parent cells. They are clones. Okay? So mitosis begins in the nucleus. So that's this center part here, this little red thing here, where the DNA must duplicate before the cell divides. So body cells, also known as somatic cells. So mitosis happens with all cells in the body, with the exception of sex cells or gametes. So your toe is part of your body. Mitosis has the word toe in it to remind you that it makes 2 cells. Mitosis begins in the nucleus. Remember, the nucleus is in control. It controls the entire process. Alright, advantages of mitosis. Quick process and makes identical copies. Disadvantage of mitosis, it makes identical copies. Yes, we did just say it was an advantage, but it's a disadvantage because when you make identical copies, there's no genetic variation. So if we were all the exact same and there was a sickness that came about, it would kill all of us instantly because there was no genetic variation. Okay? So, inside cells, I'm sure you know, there are organelles. That's these little other things outside the nucleus floating in the cytoplasm, which is the jelly of the cell. So, how does that all factor into DNA replication and mitosis? So, cells go through a cycle after they are born. They divide only when the nucleus tells them to. The nucleus decides when it's time to divide. The cell cycle is IP on a mat, which is interphase, prophase, metaphase, telophase, anaphase. Sorry, anaphase, telophase. Wow, that was bad. So interphase, prophase, metaphase, anaphase, telophase. That's the order. All right. So nucleus, all the actions begin and end with the direction from the nucleus. The nucleus is in control. It is the brain of the cell. Chromosomes in the nucleus contain the genes which control cell activities. So your chromosomes are made up of DNA, which is nucleic acid, made up of nucleotides that are found in the nucleus. Okay. Stages of the cell cycle. Cells spend most of their time in interphase. That's I. This is the phase where they perform most of their life functions. So, strange to see. Alright, the stages of mitosis are prophase, metaphase, anaphase, telophase, and cytokinesis only occurs in animal cells. So, prophase, metaphase, anaphase, telophase, cytokinesis, and interphase up the top there. Alright, so interphase, or I. Interphase is the in-between phase. So that's the period that cells have between cell divisions. Most of a cell's life is spent in interphase. So when it's time to divide, at the end of interphase, single-stranded chromosomes, also known as chromatids, replicate, make an exact copy of themselves. Alright, so we have one chromosome, or a chromatid, just chilling out there, going through interphase, and then it duplicates itself. So now we have one chromosome duplicated. So this is one chromatid, and this is its sister chromatid. Okay? And they're the exact same, and they pair up here in the middle. Okay? So chromosomes, the original chromatid, that's the one single strand, is copied, and the copy is attached like Velcro to the original at the centromere. So that's these funny dots here. They're like the Velcro holding it together. So instead of having 2 chromatids, we now have one chromosome. Right. Next, structure of a chromosome. So we have an unduplicated one, so our chromatid, or 2 that have been fused together, being duplicate, so identical sister chromatids, which make a chromosome. So 2 identical sister chromatids attach at an area in the middle called a centromere. When the cells divide, sister chromatids separate and one goes into each new cell. Okay, so the center part, don't forget, is called the centromere or primary constriction, basically where they stick together. Okay, chromatin to chromosome illusion. Chromatin, weird noodles, duplicates itself and then it coils up into chromosomes. You get more funky weird noodles. Alright, after we have interphase, we begin mitosis. So interphase is technically not part of mitosis. But mitosis has 4 phases: prophase, metaphase, anaphase, telophase. You might be going, Miss, where's cytokinesis? Technically, cytokinesis is not part of mitosis. Just these 4 phases are mitosis. Okay? Next. So 4 phases of nuclear division directed by the cell's DNA, PMAT. So prophase looks like that. We have chromosomes in the middle, spindle fibers, and spindles forming. Metaphase, remember M for middle, the chromosomes line up in the middle of the cell. Anaphase, where they start to come apart. And telophase, we have the 2 new cells. Okay, we're going to go into each of those in detail. So, prophase. In prophase, the double-stranded condensed chromosomes become visible. So here, we can see them now. The nuclear membrane disintegrates. That's why it's a dashed line going around the chromosomes. And in animals, the centriole and the spindle fibers move to opposite sides of the cell. So these two dots, this dot here and this dot here, are the centrioles, and the lines between them are the spindle fibers. Okay? They come into play later on. So, metaphase, really easy to recognize. These are all lined up in one nice long line. The chromosomes move towards the middle of the cell where they line up and prepare for division. The centrioles spit out the spindle fibers that attach to the middle, so the centromere, of the chromosomes. Now, meta is the prefix and it means middle. So these here, centrioles, have released their spindle fibers and they're now attached to the chromosomes. Right. Anaphase. During anaphase, the spindle fibers pull the chromatids apart. So now they're chromatid and chromatid. They are no longer chromosomes because they're split. The chromatids move to opposite parts of the cell. Remember, A for anaphase, A for apart. They're split apart. So each chromatid is now a single-stranded chromosome containing all the necessary genetic information. So this purple line here has the same information as this purple line. This red line and this red line, the same. Same goes for the blue and green. Okay? Telophase. Last official stage of mitosis is telophase. So cytokinesis is also part of this, but only in animal cells. In telophase, the nuclear membrane reappears around each set of chromosomes, and it forms two identical nuclei. Cytokinesis is different for plants and animal cells. So here, we have the nuclear membrane reforming, the cell being remade, and the middle, they are identical. They contain the same information. Alright, cytokinesis. Last official stage of mitosis is telophase, but in animal cells, we have one last phase, that's cytokinesis. Cyto, meaning the cytoplasm, so this is the jelly that organelles sit in, and kinesis is to split. Cytokinesis does not occur in plant cells. I'll say that again. Cytokinesis does not occur in plant cells. It's an animal cell special. Okay. Next. So, why? Because plants have a cell wall. The membrane cannot pinch in to create two separate cells. The plant cells need to build a wall in the middle of the cell in order for it to split into two. That wall is called the cell plate, and it's made of cellulose. Also, that's a pretty funny picture. Anyway, alright. Stages of mitosis. So, interphase, prophase, metaphase, anaphase, telophase, cytokinesis. This is what it looks like in animal cells, and this is what it looks like in plant cells. We draw plant cells as square and animal cells as round circles. If you're ever unsure which one you're looking at, look at the shape. Okay? Circle, animal, square, plant. Okay? So interphase, prophase, metaphase, anaphase, telophase, and then the resulting daughter cells. Right? You need to be able to know how to draw these and how to label these. Okay? Next, so mitosis produces two identical offspring. So starts off at 2N, creates 2N. 2N stands for diploid. 1N, or just N by itself, so like that, is haploid. Alright, so once again, mitosis, prophase, metaphase, anaphase, telophase. Okay? That's 2N, also known as diploid. Okay, so N equals the number of chromosomes in the sex cell. So in sexually producing organisms, such as us humans, the sex cell, also known as gametes, is the sperm and the egg. The N number, which is a haploid cell, is the number of chromosomes in each gamete. For us humans, it's 23. Each animal has their own individual amount. That's what makes them all different. Alright. 2N numbers are different. Every organism has its own specific number of chromosomes. So chromosome number has nothing to do with intelligence. There is no linkage between the two. So we have 46 chromosomes or 23 pairs. Dogs, 48. Goldfish, 94. This whole entire list here is different organisms and how many chromosomes they have or how many chromosome pairs they have. Most one is a fern. It has 1,260 chromosomes or 630 pairs. Now, that's a lot of genetic information. Okay? So, when the nucleus loses control of mitosis, we get cancer. And if it goes on long enough, that cancer becomes malignant, and yada, yada, yada. So, cancer is basically cell division gone bad. Okay? So, this is a diagram. I'm going to assume it's of an onion because that is the most common thing to take photos of. So, these two cells here are in metaphase. These two are in prophase. This is interphase, anaphase, and telophase. If given a diagram like this, you need to be able to label what cells are in what part of the process. So have a go at it. If you have the PowerPoint downloaded, you can label it like they've done here. Alright, break time. Take a break, go for a stretch. Come back and we will start meiosis. Alright, so meiosis is a form of cell division in which gametes with half the number of chromosomes are produced. So diploid, 2N, haploid, N. Meiosis is sexual reproduction. And two divisions, so we have meiosis 1 and meiosis 2. Alright, so fertilization. The fertilization of a sperm and egg to form a zygote. A zygote is a fertilized egg. So we have our egg, sperm meets egg, makes a zygote. 23, 23, 46. If you're given a question and it tells you how many chromosomes are in the gamete, when it forms a zygote, it is double the amount. Okay? Alright. Meiosis. Sex cells divide to produce gametes, so either a sperm or an egg. Gametes have half the number of chromosomes. Occurs only in the gonads, so in humans, that's your testes or your ovaries. So, male meiosis is called somatogenesis, and female meiosis is called oogenesis. Okay, meiosis is similar to mitosis with some chromosomal differences. Alright, somatogenesis. Human sex cell, 2N, is 46 chromosomes, so it's a diploid cell. Splits, makes 2N cells. So we have 23 and 23. They split again, and then we end up with 4N23 cells. So meiosis 1 makes 2, meiosis 2 makes 2 each, so we end up with 4 haploid cells. So we start off with 1 diploid and end up with 4 haploid. Okay, so meiosis in mouse testes, these are the different phases in meiosis that we're taking photos of from a mouse. Okay, so here we end up with 4 haploid cells, but we started with 1 diploid cell. Okay, parent cell, we have the first division, second division, and end up with 4 gametes. All right, interphase 1. It is similar to meiosis interphase. So chromosomes replicate, so that's the S phase of interphase. Each duplicated chromosome consists of 2 identical sister chromatids attaching to the centromeres. And the centriole pairs also replicate. So don't forget, the centriole is what creates the spindle fibers that comes into play in metaphase and anaphase. Alright. So nucleus and nucleolus are visible. So nucleus, nucleolus. So chromatin is the weird part, is this space here. Cell membrane, nuclear membrane, nucleolus. So the nucleolus is in the center of the nucleus. Alright, so meiosis 1. There are 4 phases for it. Step 1, cell division that reduces the chromosomes number by 1/2. 4 phases, prophase 1, metaphase 1, anaphase 1, and telophase 1. Alright, prophase 1. This is the longest and most complex phase. It takes up 90% of the time. The chromosomes condense. Synapsis occurs, so homologous chromosomes come together and form a tetrad. A tetrad is 2 chromosomes or 4 chromatids, so sister and non-sister chromatids. Alright, we're going to go into that in more detail. Don't forget, we are still in prophase 1. So looking at specifically the synapsis of prophase 1. Chromosome, joined together, got a tetrad, 4 chromatids. Yeah, so these are all homologous chromosomes, which means they are the same ones. So sister chromatids, tetrad. Alright, homologous chromosomes. So a pair of chromosomes, so the maternal and paternal, that are similar in shape and size. So the homologous pairs, it's now formed a tetrad, carry genes controlling the same inherited traits. So each locus or position of a gene is in the same position on homologous. So humans have 23 pairs of homologous chromosomes. So 22 pairs of autosomes, 1 pair of sex chromosomes. Okay, so we have something called a karyotype. So a method of organizing the chromosomes of a cell in relation to number, size, and type. So these are the different sizes we can get for a chromosome. So homologous chromosomes, same size. On the same spot, they share the same gene. So in this case, this is for eye color. So the locus is the position on the chromosome where the gene is found. So they have matching eye color locus and hair color locus. Okay, so from paternal, maternal. Alright, chromosomes. Each homologous set is made up of 2 homologs. So homolog, together, homologous. Homolog, together, homologous. All the way up for 22, except 23 is a little different because that's your sex pair. Okay, so in humans, the autosomes are sets 1 to 22, which means these are all the same. They pair up and they're the same size and they code for the same genes and all these. However, so autosomes code for most of the offspring traits. So looking specifically at chromosome 21, there is an extra one in this person here. What is it? Here, down here, 21. There's 1, 2, 3. So with the third chromosome for 21, that's how you get Down syndrome. So this formation here, this child is going to have Down syndrome. Now if we look here, this is chromosome pair 23. There's one X, and this chromosome here would also be an X with the same size. So this person is going to be a female because she has the extra chromosome set in spot 21 on her karyotype, is going to have Down syndrome. Oops. Okay. There. So 21, female. Alright, so in humans, the sex chromosomes are the 23rd set. So sex chromosomes, the sex chromosomes for the sex of the offspring. If the offspring has 2 X's, it's female. If the offspring has 1 X and 1 Y chromosome, it will be a male. So, female, male. So, 2 X chromosomes, female. X and Y chromosome, male. This is what they look like under a microscope. Your X chromosome is literally double the size of the Y chromosome. Okay. So, we're still in prophase 1, we have something called crossing over. So crossing over is a variation. It may occur between non-sister chromatids at the chismata. So crossing over, segments of non-sister chromatids break and reattach to the other chromatid. Chismata are the sites of crossing over. Alright, so crossing over for variation. Diagram. So, non-sister chromatids, paternal, maternal. This site here, where they're touching, is a crossing over site or chismata. So, they cross over parts here, and the end, we get some of the maternal DNAs come to the original paternal chromosome, and some of the paternal information has gone over to the original maternal chromosome. Don't forget, these 4 here are called tetrad. Right? So we have variation. Another diagram. So exchanging parts of non-sister chromatids, duplicated maternal chromosome, duplicated paternal chromosome. These 2 are sister chromatids. The 4 together are non-sister chromatids, even though they are homologous. They are non-sister because they come from different parents. Mum, dad. Mixed together, and this is the result. Some of dad went on to mum, and some of mum went on to dad. So the sister chromatids now have different genetic information. Alright, so crossing over multiplies the already huge number of different gametes produced by independent assortment. Alright, sex chromosomes. X and Y. So XX is female, XY is male. Prophase 1. So, back to this. You have your spindle fibers and aster fibers and your centrioles. They're coming out and joining to the chromosomes. Okay? Metaphase 1. The shortest phase. The tetrads align on the metaphase plate. So that is the center of the cell. So independent assortment occurs. So orientation of homologous pairs poles to pole is random. So they will randomly be situated along the pole or along the metaphase plate. Variation, formula is 2n, example 2n equals 4, then n equals 2, thus 2n squared is 4. We will get into that a little later. Okay, so metaphase 1. They line up on the metaphase plate, so either in the combination like this or a combination like this. So if you notice, this bottom pair has swapped sides. Okay. Question. In terms of independent assortment, so that's when they line up on the metaphase plate, how many different combinations of sperm could a human male produce? Well, there is an equation for that. So, answer for that. Formula 2n, because it's a diploid cell at the point in time. Human chromosome, the formula is 2 to the power of n, whatever n is. In human chromosomes, it's 2n is 46. So, n by itself is 23. So, if we punch that into the formula, it's 2 to the power of 23 is approximately 8 million combinations we can get from how they line up on the metaphase plate. Okay? Anaphase 1. So homologous chromosomes separate and move towards the poles. Sister chromatids remain attached at their centromeres. Right? So anaphase 1, these guys are still attached, the sisters are still attached, but they're just moving away to opposite sides of the cell. Right? Telophase 1. Each pole now has a haploid set of chromosomes. Okay? And cytokinesis occurs and the 2 haploid daughter cells are formed. Telophase 1. Hello. Splitting happens. This is now a haploid. This is also a haploid cell. Okay. And when they finish splitting, once the cytokinesis has gone through completely, we now have 2 haploid cells that are genetically different. All right, meiosis 2. There is no interphase 2. Okay? Or if there is, it's extremely short and there's no DNA replication. Meiosis 2 is similar to mitosis. Okay? So we have prophase 2, same as prophase and mitosis. Information is condensed. The spindle fibers form, centrioles are formed. Start attaching to the chromosomes. I mean, metaphase 2, same as metaphase in mitosis, line up along the metaphase plate, getting ready to be split apart. Anaphase 2, same as anaphase in mitosis, sister chromatids separate, boop, 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 cut in half. Alright, and then telophase 2, same as telophase in mitosis. The nuclei reform, cytokinesis occurs, and the 4 haploid daughter cells are produced in meiosis. Meiosis has 4 haploid cells. Mitosis has 2 diploid cells. Okay? So, haploid cells, gametes, so your sperm or your egg. Alright, telophase 2. Pulled apart, end up with 4 different cells. So the genetic information in each one of these cells is different. That is important to note. Mitosis has clones and they're identical. Meiosis has different cells. Different genetic information. Each of these cells are different. Right. Meiosis. Diagram. So, start off with a sex cell, 2, n equals 4. Then we split into n, there's 2, and then n2. So all of these are haploid. Start off with a diploid cell, end up with 4 haploid cells. Alright. Variation. So, important to population as the raw material for natural selection. Question. What are 3 sexual sources of genetic variation? Answer to that is crossing over. So that happens in prophase 1. Then we have independent assortment, metaphase 1. So depending on which side of the metaphase plate the chromosomes line up on, and then random fertilization. Variation is good. It's what makes us all individual and unique. It changes the way we look from our siblings to our parents and all the rest of it. All right, next question. A cell containing 20 chromosomes, so it's diploid, at the beginning of meiosis would, at its completion, produce cells containing how many chromosomes? Pause it, answer the question. Answer should be 10 chromosomes because it is haploid cells. Diploid is 20, so haploid is 10. Alright, fertilization. The fusion of a sperm and an egg form a zygote. A zygote is a fertilized egg. So our egg is N23, our sperm is N23, and our zygote is 2N of 46. Question. A cell containing 40 chromatids, not chromosomes, chromatids, that's chromatid is half of a chromosome, at the beginning of meiosis would, at its completion, produce cells containing how many chromosomes? Pause. Answer the question. Come back. Our answer is 10 chromosomes. Alright. Break time. Go. Bathroom. Have something to drink, have something to eat. Come back. And we're going to do traits and genes. Alright, so key concepts. So genes encode proteins that produce a diverse range of traits. A gene is a piece of DNA that tells a cell to make a certain protein. Each gene has a locus, a specific position on a pair of homologous chromosomes. If you don't remember what homologous means, go back about 5 minutes while we go through that. Alright, so homologous chromosomes, so the alleles, so the gene, the variation of a gene, so you either have wrinkled or wrinkled. So each allele is an alternative form of a gene. Each parent donates one allele for every gene. For shape, there is a wrinkled allele and a round allele. They received one allele from each parent. So here we have homozygous alleles, homozygous meaning the same, heterozygous meaning different. So this person here has 2 wrinkled alleles. This person here has 1 wrinkled and 1 round. So heterozygous alleles are different from each other, and homozygous alleles are identical to each other. Alright, on to genotype. For homozygous, so a pure combination. So when both letters are the same. So going back to our Punnett squares, 2 capital T's or 2 lowercase t's. So it's either pure dominant or pure recessive. Heterozygous is when we have a hybrid when the letters are different. So either small and big, small and big T or small and big X, whatever it may be. So dominant alleles are always uppercase and recessive alleles are always lowercase. Alright, so Mendel was the dude who observed genetic variation in traits. He came up with this rule, the rule of dominance. So when a dominant trait is present, the recessive trait is hidden. Okay, so gene influences the development of traits. All of an organism's genetic material is called the genome. A genotype refers to the actual genes, and a phenotype is a physical expression of a trait. So for a genotype, it might be uppercase B, lowercase b, and when that gene is expressed, it might show that, I don't know, if it's in mice, it might have brown fur. Okay? So genotype, the gene combination of an organism, consists of 2 alleles. For example, uppercase, 2 uppercase P's, an uppercase and a lowercase p, or 2 lowercase p's. Alright, that is a Punnett square. So the phenotype is the appearance. So the genotype determines the phenotype. Oh, go back. Alright. So, for this diagram here, we have yellow peas and green peas. Yellow peas is dominant and green peas are recessive. So, when we have our Punnett square, because yellow is dominant, that's what's expressed. So the genotype is uppercase Y, lowercase y, and the phenotype is a yellow pea. For the green one here, the genotype is 2 lowercase y's, and the phenotype is a green pea. Okay? So probability is the likelihood of an event. So probability is expressed as a percentage or a fraction. So with the genotype ratio is the ratio of genes that will appear. Phenotype ratio, the ratio of phenotypes that will appear. We can find the probability of a certain genotype or phenotype by constructing a Punnett square. So when a dominant allele always gets its way, so the dominant phenotype is present no matter its partner, it is called complete dominance. So example, uppercase and lowercase p will code for a purple flower, as well as 2 uppercase p's. Sometimes dominant traits don't completely get their way. The alleles compromise. This results in a phenotype that is a mix of both parents. This is incomplete dominance. So for example, if we had an uppercase P and a lowercase p, and that resulted in a light purple flower, that is an example of incomplete dominance. Okay? So in a few instances, both alleles need to be expressed. That is co-dominance. For example, a mother chicken is black and the father chicken is white, but the baby chick comes out black and white. Alright, most traits occur in a range and do not follow simple dominant recessive patterns, like with the P's. Alright, so traits are inherited through genes. Genes are on chromosomes. 1 chromosome is inherited from each parent, as we talked about in meiosis, and we have 2 copies of each chromosome and thus 2 copies of each gene. So if one of them is faulty, we have a backup, unless it is on the 23rd pair and you're a dude. Then you don't have a backup and you're kind of stuffed. Alright, so different versions of genes are alleles. So for each gene you might have 2 different alleles or 2 of the same allele. Combinations of alleles determine what traits you have. So if you can roll your tongue, you carry a dominant allele. It's either a dominant and a recessive or 2 dominant genes. If you cannot, you have 2 recessive genes. So if you inherit both alleles, the dominant one is expressed, in this case here. However, dominant does not mean that it is more common. It just means if you were to put it against a recessive gene, the dominant gene would more than likely be expressed. Alright, break time. We're going into pedigrees. Come back and we will get cracking. Alright, so what is a pedigree? A pedigree is a family tree that shows how a trait is passed down from generation to generation. Like this. Alright, some pedigree symbols. We have male, female, mating, parents and children, in order of birth. So eldest is to the left and youngest is right. We don't need to worry too much about this one here unless it's the sex unspecified. So that's when the woman is pregnant and she hasn't given birth yet and they don't know if it's going to be a male or a female. So we put as a unspecified. All right. Number of children of sex indicated. Affected individuals are shaded. Heterozygous or autosomal recessive, which means they carry the gene. Carrier of sex-linked recessive, we don't worry about that one. Death, they're dead. We don't worry about that one, or that one, or that one. Alright, so on this one here, squares indicate males, so count them. There are 7 males. 1, 2, 3, 4, 5, 6, 7. 7 males in this pedigree. Alright, circles indicate females. Count them. You should have 7 females in this pedigree as well. Alright, shaded shapes represent individuals that have the trait that we're looking for. And there are 2 individuals that are affected in this pedigree here. They're both males. So, mating between individuals are shown by horizontal lines. So, all of these people here mated and produced offspring. Alright, so with pedigrees, how many generations are shown here? On the side here, there are Roman numerals. So generation 1 is in this line, generation 2, and generation 3. So 3 generations here. Alright, vertical line form relationship. So vertical form relationship line, children. So these 2 had offspring, and that's the line. So this is the child, this is one of the children, this is one of the children. And these guys had offspring here. Okay. Alright. How many children do parents from generation 1 have? So follow the lines down. You should be able to count. And your answer should be 3 children. Alright. How many children in generation 3 have the trait? So we follow down. Generation 3. This one here is the only one with the trait. So there is 1 person with the trait. Alright. Another pedigree. So how many people in the family have the trait? Count them. Your answer should be 7 people have the trait that we're looking at. Alright. Does the individual I1, so generation 1, number 1, have the trait? So what is the sex of the individual? He's male because he's a square. Does he have the trait? Yes, because he's shaded in blue. Alright. So list the individuals in generation 3 that have the trait. So generation 3, that one, and that one. So they are triple I 1, triple I 6, and triple I 8. So when you're asked which individual is who in the pedigree, you first of all numbered them from left to right, 1 through to whatever number they go up to, and then what generation they're in. So if you were to ask what this one here, with what? If you were to ask who this would be in the pedigree, you would say it is 3i4, 3i-4. That's who that person is. Alright, so next, how many children did the couple from generation 1 have? So here's generation 1. Count the lines going down, and that tells you how many kids they had. They had 5 kids. Alright, pedigree 2. What is the relationship between individual 2i8 and 3i9? So 2i8 is here, and 3i9 is here. So 8 and 9. 8 is female because she's a circle, and 9 is also female because she's also a circle, so it must be mother-daughter. Right, so pedigree and traits. Pedigrees are most useful when analyzing a family's history of genetic diseases and traits, but can also predict if future family members will inherit disease or trait using a pedigree. We are going to go through some examples. Alright, so example 1. Pedigree below shows the inheritance of handedness in humans over 3 generations. The allele for right-handedness is dominant over left-handedness. Shaded individuals below are recessive for handedness. So these people here that are shaded in are all left-handed. Okay, and then we can do a Punnett square with right-handed and left-handed people because we have that information as well. Alright, so first question, how many generations are there? Next one, how many individuals are there? How many individuals are left-handed? And last one, which individual in the 3rd generation are right-handed? Right? Go ahead and answer them. Alright, so we have 3 generations, 4 people are left-handed. In 3rd generation, it's 3i-1 and 3i-3. Right, next. What is the genotype and phenotype of individual I2? So I2 is here. Genotype, now we know this is recessive, and the recessive gene was lowercase r, so it's lowercase r, lowercase r. Phenotype, how is that expressed? It's expressed because she is left-handed. Alright, so if the father, I1, up here, is heterozygous, what is the genotype of his 2 children? Go, 1, 2. So he's heterozygous, which means he has an uppercase R and a lowercase r, which means this one here is lowercase r, and this one here is uppercase R, lowercase r. Ta-da! There should be your answers there. Alright, example 2. Huntington's disease is a dominant disorder. So what is the relationship between individual 3i1 and 3i2? 3i1, so these 2 people here. What's your answer? Your answer should be brother and sister. Alright, next. Relationship between 2i3 and 3i4. So here and here. Take a crack at it. 3, 2, 1. Aunt and nephew. Okay. Oh. So aunt and nephew. Female, male, aunt and nephew, because these 2 are sisters. And that's why we have aunt and nephew. Where's it? Spacebar. There it is. Alright, how many individuals in generation 2 do not have Huntington's disease? So this is generation 2. So the ones who don't have Huntington's disease are unshaded. Your answer should be 3 people. The 3 that aren't shaded. Right? So based on traits of IV5 and IV6 and IV7, what must the genotype of 3-8 be? Right, so 3-8, what's this person's genotype? Based on 1, 2, 3, what is that genotype going to be? I think that's incorrect. It's meant to be asking what this one here is, because we can tell that's clearly recessive. So this one here, this female here, has a dominant and a recessive gene. Sorry, has 2 dominant genes. Sorry, no. No, wait, dominant. Has a dominant and recessive gene. So, this person here, these are all dominant and they aren't. So, these all have dominant and recessive genes and are not affected by it. Okay. Next, if the individual IV7 mated with heterozygous individual, what is the percentage of their offspring having Huntington's disease? To answer a question like this, you need to do a Punnett square. So, genotype of IV7 is HH. How is it HH? So, Punnett square, and 50% has the disease. So that's these 2 here will have Huntington's. Alright. One note on sex-linked pedigrees. For sex-linked traits, female carriers are always represented as a circle halfway shaded, even though they do not express the recessive trait. So this is what a carrier looks like. Half colored in. Alright, this here is a massive pedigree of a royal family. If you look down here, you will see the British royal family. So there's Prince William and Prince Henry, Princess Diana and Prince Charles. And this, I believe, is for hemophilia. Sorry, hemophilia, not hematophilia. So hemophilia, so that's basically a condition where your blood cannot clot. So if we look all the way from back up here, we are starting with King George III. And then his wife had Duke Edward of Kent, Edward Duke of Kent had Queen Victoria, and they were cousins. Prince Albert and Queen Victoria were cousins, and then they had a bunch of children. They had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 kids. I'm sorry, they had 10 kids? Anyway, so Queen Victoria here is a carrier for hemophilia. Sorry, hemophilia. Why did I keep adding the T? Anyway, Queen Victoria is a carrier for hemophilia, and along the line she had 1 kid, Leopold, who had hemophilia. Both Alice and Beatrice are carriers though. Alright, so Beatrice and Prince Henry procreated, and they had 1, 2, 3, 4 children, 2 of which had hemophilia, Maurice and Leopold. What's the name Leopold? Anyway, they also had Queen Eugenie, who is a carrier for hemophilia. Alright, she married Alfonso, King of Spain, and they had 1, 2, 3, 4, 5, 6 children, 2 of which have hemophilia. And then we have 2 question marks here, which has no evidence of hemophilia. And then Juan, I think, had a son or became King Juan Carlos. Okay, so each of these shaded squares here show someone who has expressed hemophilia. And if you notice, they are all male, which tells us that hemophilia is an X-linked gene. All the females are carriers because they are all half shaded. There is no female here that is shaded to show that they have hemophilia. Alright, and that's the end. Right, I hope you found that useful. If there's anything that I did not go over or you would like further questions on, please email me or ask me in class or leave a comment down below and I will try to get to it, okay? Good luck with your exam. I'm sure you'll be fine. Bye-bye.